Sensation of taste. Chapter 176. Taste buds. Sense organs for taste or gustatory sensation are the taste buds. Taste buds are ovoid bodies with a diameter of 50 to 70. In adults, about 10,000 taste buds are present, and the number is more in children. In old age, many taste buds degenerate and the taste sensitivity decreases. Situation of taste buds. Most of the taste buds are present on the papillae of tongue. Taste buds are also situated in the mucosa of epiglottis, palate, pharynx, and the proximal part of esophagus. Types of papillae located on tongue: Filiform Papillae, Fungiform Papillae, Circumvallate Papillae, Filiform Papillae. Filiform papillae are small and conical-shaped papillae, situated over the dorsum of tongue. These papillae contain less number of taste buds, only a few. Fungiform papillae. Fungiform papillae are round in shape and are situated over the anterior surface of tongue near the tip. Numerous fungiform papillae are present. Each papilla contains moderate number of taste buds, up to 10. Circumvallate papillae. Circumvallate papillae are large structures present on the posterior part of tongue and are many in number. These papillae are arranged in the shape of V. Each papilla contains many taste buds, up to 100. Structure of taste bud. Taste bud is a bundle of taste receptor cells, with supporting cells embedded in the epithelial covering of the papillae, Fig 176.1. Each taste bud contains about 40 cells, which are the modified epithelial cells. Cells of taste bud are divided into 4 groups: type of cells in taste bud. Type I cells or sustentacular cells, type 2 cells. Type 3 cells. Type 4 cells or basal cells. Type I cells and type 4 cells are supporting cells. Type 3 cells are the taste receptor cells. Function of type 2 cell is unknown. Type I, 2 and 3 cells have microvilli, which project into an opening in epithelium covering the tongue. This opening is called taste pore. Neck of each cell is attached to the neck of other. All the cells of taste bud are surrounded by epithelial cells. There are tight junctions between epithelial cells and the neck portion of the type I, 2 and 3 cells, so that only the tip of these cells are exposed to fluid in oral cavity. Cells of taste buds undergo constant cycle of growth, apoptosis, and regeneration. Pathway for taste. Receptors. Receptors for taste sensation are the type 3 cells of taste buds. Each taste bud is innervated by about 50 sensory nerve fibers, and each nerve fiber supplies at least 5 taste buds through its terminals. First-order neuron. First-order neurons of taste pathway are in the nuclei of 3 different cranial nerves, situated in medulla oblongata. Dendrites of the neurons are distributed to the taste buds. After arising from taste buds, the fibers reach the cranial nerve nuclei by running along the following nerves: Fig 176.2 chorda tympani, fibers of facial nerve, which run from anterior 2/3 of tongue. Glossopharyngeal nerve fibers, which run from posterior 1/3 of the tongue. Vagal fibers, which run from taste buds in other regions. Axons from first-order neurons in the nuclei of these nerves run together in medulla oblongata and terminate in the nucleus of tractus solitarius. Second-order neuron. Second-order neurons are in the nucleus of tractus solitarius. Axons of second-order neurons run through medial lemniscus and terminate in posteroventral nucleus of thalamus. Third-order neuron. Third-order neurons are in the posteroventral nucleus of thalamus. Axons from third-order neurons project into parietal lobe of the cerebral cortex. Taste center. Center for taste sensation is in opercular insular cortex, i.e. in the lower part of post-central gyrus, which receives cutaneous sensations from face. Thus, the taste fibers do not have an independent cortical projection. Primary taste sensations. Primary or fundamental taste sensations are divided into 5 types: sweet, salt, sour, bitter, umami. Man can perceive more than 100 different tastes. Other taste sensations are just the combination of two or more primary taste sensations. Combination of taste sensation with other sensations. Sometimes, taste sensation combines with other sensations to give rise to a different sensation. For example, combination of taste, smell, and touch senses gives rise to sensation of flavor. Combination of taste with pain gives rise to sensation of ginger. Discrimination of different taste sensations. Earlier, it was believed that different areas of tongue were specialized for different taste sensation. Now it is clear that all areas of tongue give response to all types of taste sensations. Usually, in low concentration of taste substance, each taste bud gives response to one primary taste stimulus. However, in high concentration, the taste buds give response to more than one type of taste stimuli. It is also clear now that each afferent nerve fiber from the taste buds carry impulses of one taste sensation. Taste sensations and chemical constitutions. Substances causing sour or salt tastes are mostly electrolytes. Bitter and sweet tastes are caused by electrolytes or non-electrolytes. Sweet taste. Sweet taste is produced mainly by organic substances like monosaccharides, polysaccharides, glycerol, alcohol, aldehydes, ketones, and chloroform. Inorganic substances, which produce sweet taste, are lead and beryllium. Salt taste. Salt taste is produced by chlorides of sodium, potassium, and ammonium, nitrates of sodium and potassium. Some sulfates, bromides, and iodides also produce salt taste. Sour taste. Sour taste is produced because of hydrogen ions in acids and acid salts. Bitter taste. Bitter taste is produced by organic substances like quinine, strychnine, morphine, glucosides, picric acid, and bile salts, and inorganic substances like salts of calcium, magnesium, and ammonium. Bitterness of the salts is mainly due to cations. Umami. Umami is the recently recognized taste sensation. Umami is a Japanese word, meaning delicious. Receptors of this taste sensation respond to glutamate, particularly monosodium glutamate, message, which is a common ingredient in Asian food. However, excess message consumption is proved to produce Chinese restaurant syndrome in some people taking Chinese food regularly. Common symptoms are headache, flushing, sweating, perioral numbness, chest pain. In severe conditions, airway swelling and obstruction and cardiac arrhythmia occur. Threshold for taste sensations. Sweet taste: sugar, 1 in 200 dilution; salt taste. Sodium chloride, 1 in 400 dilution; sour taste. Hydrochloric acid, 1 in 15,000 dilution. Bitter taste: quinine, 1 in 2 million dilution. Bitter taste has very low threshold and sweet taste has a high threshold. Threshold for umami is not known. Taste transduction. Taste transduction is the process by which taste receptor converts chemical energy into action potentials in the taste nerve fiber. Receptors of taste sensation are chemoreceptors, which are stimulated by substances dissolved in mouth by saliva. The dissolved substances act on microvilli of taste receptors exposed in the taste pore. It causes the development of receptor potential in the receptor cells. This in turn is responsible for the generation of action potential in the sensory neurons. Taste receptor. Generally, taste receptor is a G-protein coupled receptor, GPCR. It is also called G-protein gustatory. However, several other receptors are also involved in taste sensation. Transduction mechanism is different in each taste receptor cells. Sweet receptor. Receptor for sweet taste is GPCR. The sweet substances bind to receptor and cause depolarization via cyclic AMP. Salt receptor. Receptor for salt taste is called epithelial sodium channel, ENAC. It acts like ENAC receptors in other parts of the body. When sodium enters, this receptor releases glutamate, which causes depolarization. Sour receptor. Sour sensation also has the same ENaC receptor. The proton, hydrogen, enters the receptor and causes depolarization. It is believed that besides ENaC, other receptors such as hyperpolarization-activated cyclic nucleotide-gated cation channel, HCN, also are involved in sour sensation. Bitter receptor. Bitter receptor is a GPCR. In bitter receptor, the sour substances activate phospholipase C through G proteins. It causes production of inositol triphosphate, IP3, which initiates depolarization by releasing calcium ions. Umami receptor. Umami receptor is called metabotropic glutamate receptor, mGlu-R4. Glutamate causes depolarization of this receptor. Exact mechanism of depolarization is not clearly understood. Activation of umami taste receptor is intensified by the presence of guanosine monophosphate, GMP, and inosine monophosphate, IMP. Applied physiology, abnormalities of taste sensation. Aguzia. Loss of taste sensation is called aguzia. Taste buds in anterior 2/3 of the tongue are innervated by the chorda tympani branch of facial nerve. Chorda tympani nerve receives taste fibers from tongue via lingual branch of mandibular division of trigeminal nerve. So, the lesion in facial nerve, chorda tympani or mandibular division of trigeminal nerve causes loss of taste sensation in the anterior 2/3 of the tongue. Lesion in glossopharyngeal nerve leads to loss of taste in the posterior 1/3 of the tongue. Temporary loss of taste sensation occurs due to the drugs like captopril and penicillamine, which contain sulfhydryl group of substances. Hypogeusia. Hypogeusia is the decrease in taste sensation. It is due to increase in threshold for different taste sensations. However, the taste sensation is not completely lost. Taste blindness. Taste blindness is a rare genetic disorder in which the ability to recognize substances by taste is lost. Dysgeusia. Disturbance in the taste sensation is called dysgeusia. It is found in temporal lobe syndrome, particularly when the anterior region of temporal lobe is affected. In this condition, the paroxysmal hallucinations of taste and smell occur, which are usually unpleasant.